Hello, grade 12 students. Here we move for genetics for the chapter 3, genetic variations and the polymorphism. So chapter 3, genetic variation and polymorphism, we're going to talk about document 2, about the mutations and the multiple allele, pages 60 and 61 on your book. So first, what is a mutation? Mutation is the change in the nucleotide sequence of the DNA. This mutation is going to be permanent change in the DNA sequence, meaning that it's going to be permanent, it's going to be all over the generation. Mutation can be of two types, either in the germ cells or in the somatic cells. The mutation in the germ cells, they are going to be transmitted to the next generation, and also they will give rise to inherited diseases, whereas the mutation in the somatic cells, they are not transmitted to the next generation, meaning that they are not going to give rise to inherited diseases, but they are important in the causation of the cancers and some congenital malfunctions. The agents that can cause mutation, they are referred to as mutagens. Mutagens, there are very different types, including X-ray, the gamma ray, UV ray, and many others. Let's move now to the different types of mutation that exist. We have mutations, either they are frame shift mutations or they are point mutations as substitution. So frame shifting mutation can be divided into either deletion or insertion. As their name indicates, deletion is the deletion of one of the nucleotide, or insertion is inserting one of these nucleotides. These nucleotides, they are the A, T, C, and G, where A and T, they are complementary, C and G, they are complementary. So deletion is deleting one of these nucleotides, whereas insertion meaning that adding one or these nucleotides. Whereas point mutation can be silent. The meaning of silent, it doesn't cause any change, it doesn't cause any difference in the amino acid sequence. Why? We will determine this in the coming slides. Also, the point mutation can be missense mutation. It can change the nucleotide sequence, it can change the amino acid sequence, and it gives different amino acids, or the nonsense mutation, or the stop. They code for the stop codon. Point mutation occur when only one base of these DNA is altered or changed. The effect can be either silent mutation, missense mutation, or nonsense mutation. Silent mutation. There is no detectable effect, no effect. There is no change in the sequence of the amino acid. For example, changing the codon. What the meaning of the codon? Each one of these, we said that is called a nucleotide. Every three is called a codon or a triplet. So changing a nucleotide from CGA to CGG. So we substitute the A with the G. This substitution, it does not affect the protein sequence, since both codons CGA and CGG, both of them, they give the amino acid arginine. So this is called a silent mutation, where more than one codon, they can give the same amino acid, and this is known by redundancy. So redundancy, more than one codon, they can give the same amino acid and occurs in the genetic code table. Missense mutation. Missense, there is a different amino acid. There is a different amino acid that is formed during to this mutation. This missense mutation, it causes the appearance of a different amino acid, as we said, in the site in the protein molecule. For example, the normal blood cells to sickle cells or sickle red blood cells. This is the normal round shape. The sickle cell, it's due to one of these nucleotides, where GAA to GUA or GAG to GUG. they change from the glutamic acid to the valine. So this will lead to the appearance of the sickle cell red blood cells. They have the sickle cell, sickle shape. So this is a missense mutation, or this is the effect of the missense mutation. Nonsense. Nonsense mutation, it leads to the appearance of the stop codon. So this stop codon will lead to the stop of the synthesis of the amino acid. So this mutation, it leads to the conversion of the amino acid into a stop codon, where the synthesis of the amino acid or the synthesis of protein will stop here. Example about this, one type of thalassemia in codon 17 of the beta chain is changed from UGG to UGA. So one of these nucleotides from G into A. So this will result in the change of the codon from tryptophan to a stop codon, or scale the nonsense codon. So one nucleotide is changed, so this will lead to the appearance of the stop codon, which is the UGA. Do you want to study the codons, and do you have to study the genetic code table? Sure not. This genetic code table is going to be given in the exam. Frame shifting mutations. Frame shifting mutations, these mutations, they occur when one or more base pairs, they are either inserted or they are deleted from the DNA. Either by insertion they cause insertion mutations, or whenever they are deleted they cause deletions mutations. So this is the frame shifting mutation, where ATG goes for MAT, GAA goes for G allele. So deleted one of these G is deleted, so it's going to be shifted. So G is going to move here. So this is called shifting. So the amino acid sequence is going to be shifted in this direction. So it's going to be AAG, and then CAC, and then we have GT alone. This will lead to the change in the amino acid sequence. This will give lysine, and this will give histidine instead of alanine. Frame shifting mutations, the addition or even the deletion of a single base, they have many more profound consequences than does the substitution. Let's take this example. This example is showing the effect of the frame shifting mutations. Changing of one letter in this sentence, the cat saw the dog. Changing of one letter, so it will lead to the bat saw the dog, still have a similar meaning. Or the cat saw the hug, the D is substituted by H. Or the cat sat the dog, so sat instead T instead of W. So this would have the same meaning. Why? If we lose the C, so here the C is deleted here in this case, so it becomes the ats, out, head, ug. So loss of C says deletion. So frame shift mutation by deletion, no meaning. That's how it's a higher effect. Or by insertion, we insert the letter M between C and A, so inserted letter M here, so it can become the CMA, TSA, WTH, ADOG, so no meaning. So this is by insertion, frame shifting mutation by insertion. If frame shifting mutation takes place either in insertion or in deletion, as we said, this will lead to the change in the sequence of the amino acid where after the mutation. Okay, pay attention, after the mutation. As I said, the codon is composed of 3 nucleotides. Each codon will code for one amino acid, and we have more than one codon. They can code for the same amino acid. This is what we call by redundancy. There are 3 stop codons. These 3 stop codons, you have to study them: UAG, UGA, and UAA. They are the stop codons. So you have to study only the 3. With the point mutation by substitution, it can be silent, missense, or nonsense. Let's start with the missense. Missense mutation in one codon causes a change in one amino acid. This is called the missense. Whereas the nonsense stop codon is formed. So we have a stop codon. These stop codons, as we said, they are the UAG, UGA, and UAA. They are called the stop codons. Whereas silent mutation, it causes a no change in the amino acid sequence, where the change will lead to the same amino acid. So here are the three types of the point mutation by substitution: either missense, where it leads to the change in the amino acid; nonsense; or silent, where there is no change. So nonsense, no way to continue. So always keep it in your mind. Nonsense, no way to continue. So we need the stop codon. So this is the nonsense mutation. And the missense, they will change the sense. So we'll change the amino acid sequence. DNA and RNA. What are the differences between them? DNA is double stranded, whereas the RNA is single stranded, only one strand. The nucleotides in the DNA, they are A, T, C, and G, whereas the nucleotides in the RNA, they are A, U, C, and G. So the T is replaced by U. Sugar in the DNA is deoxyribose, where in the RNA it's ribose. DNA is long-lived, has long life or long-term life, whereas the RNA is short-lived. DNA is stable chemically, where the RNA is unstable. DNA is found in the nucleus, whereas the RNA can be found in the nucleus or in the cytoplasm. Let's now talk about the MHC, major histocompatibility complex. Major histocompatibility complex are molecules that are found on the surface of all nucleated cells. Nucleated meaning that they have nucleus. So red blood cells, they are anucleated cells. They don't have nucleus. They give individual their biological identity. So MHC, they are found of two classes, either MHC class 1 or MHC class 2. These are in Roman number 1 and number 2. MHC class 1, they are divided into class A, B, and C, while class 2, DP, DQ, and DR. So these are the MHC classes, class 1 and class 2. Subclasses, class 1, ABC, and class 2, ADBQ, DQ, DR. MHC, they are coded by 6 genes. They are found on chromosome number 6. MHC molecules, they differ from one person to another. All the cells of the same individual, they have identical MHC, and MHC molecules, they are co-dominant with respect to each other, meaning that both of them, they are co-expressed. You have to know that your MHC is composed from paternal and maternal origin. Paternal from your father and maternal from your mother. So you have your MHC, it's half of that from your father and half from your father. So it's called the maternal and paternal MHC. Both of them, they are expressed. Here we move to this application. In this application, they said consider the sequence of the DNA molecule shown below. Here we have the sequence GCA, TGC, ATC, TGC, CAT, and TGC. This is the non-template. Non-template also is called the non-transcribed. Also, it's called the coding sequence. Number 1, they write the sequence of the mRNA molecule shown, and 2, how may relate the mRNA to the template to the non-template. First of all, this is the non-template. This is the non-transcribed, and this is the coding sequence. So first of all, you have to know that non-template is called the non-transcribed, and this is the coding, while the template strand is called the transcribed, and it's the non-coding. If I want to write the template for this, the template for the non-template, I have to write the complementary. So the complementary of G, it's C. Complementary of C, it's G. Complementary of A, it's T. Complementary of T, it's A. Complementary of G, it's C. Complementary of C, it's G. Complementary of A, T. T, it's A. C, it's G. T, it's A. G, it's C. C, it's G. C, it's G. A, it's T. T, it's A. T, it's A. G, it's C. And C, it's G. So this sequence here is called the template, the transcribed, and also is called the non-coding. Okay, so here, if I want to write the complementary, it should be complementary. So G, the complementary is C. C, complementary is G. A, the complementary is T. T, the complementary is A. G is the complementary is C. C, it's a G. T, A, it's T. T, it's A. C, it's G. T, it's A. G, it's C. C, it's a G. And then we have C, it's a G. A, it's T. T, it's A. T, it's A. G, it's C. And C, it's G. So this is the complementary for the template. So here you have to keep it in mind that the non-template is called the non-transcribed, it's called the coding. So you have to study these three names: non-transcribed, non-template, coding. While the template is called the transcribe and it's the non-coding. So if you wanna find out the sequence of the mRNA as shown here, we can solve it in two ways. Either I can use the non-template, the non-transcribed, the coding. So mRNA is identical to the non-template, but instead of T is replaced by U. So this is the non-template. It's identical to mRNA, but instead of T is replaced by U. So the first GCA is GCA. TGC, the T is replaced by U. So UGC. ATC, A, it's A. T is replaced by U, and C, it's the same. T is replaced by U, and we have GC, GC. C, C, A, A. T is replaced by U. Also T is replaced by U, G, G, C, C. This is the mRNA. Or, if when I use the template, I should make it complementary. But instead of T, it's going to be U. So the complementary of C is to G. The complementary of G, it's C. The complementary of T, it's A. While the complementary of A, it's T. But in the mRNA, we don't have C. We don't have T. We have U. The complementary of C, it's G. The complementary of G, it's C. The complementary of T, it's A. While the complementary of A, it's not T, it's U. In the mRNA, we don't have T. G, the complementary, it's C. A, the complementary is U. C, G, G, C, G, it's C, T, it's A, and A, it's U. A, it's U, C, it's G, and G, it's C. Someone is going to ask me when I want to start with the non-template, when I have to start with the template. This depends on the exercise. If they gave you the non-template and they need the mRNA, it's the same. Instead of T is replaced by U. So the mRNA strand is the same as the non-template, which is the non-transcribed, which is the coding, but the T is replaced by U. While the mRNA strand is complementary to the template strand. Okay, as shown here. Here we did it a complementary, but instead of T is replaced by U. Let's now move to the document for detection of genetic polymorphism. Talk about the restriction enzymes. What are the restriction enzymes? They are DNA cutting enzymes. They are scissor enzymes. Each enzyme, it recognizes one or few target sequence. They have a specific target sequence as shown here. It has a specific target sequence here, so it cuts in a specific sequence and cuts the DNA at this sequence or even near to this sequence. Many restriction enzymes, they make staggered cuts, meaning that producing ends with single DNA overhangs. One of these restriction enzymes is the ECHO-R1. ECHO-R1 has a specific target sequence to cut into the sequence. The number of fragments is the number of restriction sites plus 1. What's the meaning of that? For example, I have a restriction enzyme. This restriction enzyme, it has a specific sequence. It has a specific site or a target sequence. This target sequence is repeated 3 times. How many fragments? So it's going to cut 3 times. So the number of fragments, number of pieces is going to be the number of the restriction sites plus 1. How many times it will cut? Plus 1. For example, it has 2 restriction sites, so then it will cut 2. So the number of pieces will be 3 pieces or 3 fragments. The extraction enzymes, they cut double-stranded DNA at specific sites or sequences. Ligase makes ligation. It joins the two DNA molecules. Gel electrophoresis is talking about the zemogram. It's used to determine whether an allele is normal or abnormal. How? By determining the genotype of the individual. So to allow also to detect the length of the DNA molecule. For example, if we have one, this is called a band, and this is called a band, also this is called a band, and this is called a band. But this one is called thick band. What's the meaning of this? One band meaning that it has this allele, and maybe it has the other allele, while one thick band meaning that this person is homozygous. Well, the meaning of this, it has 2 copies of this allele. So it has 2 copies of this allele is called the homozygous. So this is the significance of this thick band. So the thick band, as a note, the zemogram represent 2 copies of the allele, meaning that the person is a homozygous, has 2 identical alleles. Let's move to document 5, genetic identity of the individual. Here we talk about FISH technique. FISH technique is used to locate a gene on a chromosome. FISH technique is called fluorescent in situ hybridization. First of all, you have to sequence a gene. Sequencing of a gene, determining its sequence. So this is the sequence ATG, CCA, ACC, GTC, AGG, and so on. Then we have to prepare a probe. This probe contains about 10 to 12 nucleotides. It's single-stranded, it's radioactive, and it's complementary to the sequence. So here, as you see, it's radioactive, meaning that it emits light. So here, ACC, complementary of A, it's T. C, it's G. C, it's G. Then G, it's C. T, it's A. C, it's G. Then A, it's T. G, it's C. G, it's C. And T, it's A. Here we have radioactivity in the star. So here we sequence and we make a probe, a single-stranded. This probe will bind. It's complementary to the sequence of the discovered gene as it's seen here. Then, for 3, we have to unwind the 2 DNA strands. Partially on the chromosomes, we have denaturation of the DNA. The DNA is denatured. And then we have to add the probe to the tube containing the chromosomes. Then we have to examine the chromosomes, where the fragments of the chromosomes, where radioactivity is deleted, is going to be the location of the gene. And this is the aim of the in situ hybridization. Fluorescent in situ hybridization is to locate a gene on the chromosome. So where the radioactivity is deleted is going to be the location of this gene. This is one of the most important techniques and most significant techniques in the world of genetics: fluorescent in situ hybridization, FISH technique, in order to locate a gene on the chromosome, where we have to sequencing of a gene, preparing a probe, then unwinding the 2 DNA strand, partially on the chromosome, where it's called denaturation, adding the probe to the tube containing the chromosomes, examine the chromosomes, the fragment of the chromosome where the radioactivity is deleted is going to be the site, or it's going to be the location of the gene. Probe can be in 2 different models, either in monolocus or multilocus. Monolocus probe, it binds to one and only one specific site. It's called specific sequence. It's repeated only one time, where it's used in the gel electrophoresis. Or it can be multilocus. What the meaning of multilocus? It binds to more than one site. It binds to more than one specific repeated sequence. This sequence is going to be repeated more than once in the genome, the DNA. Used in DNA fingerprint, the multilocus, it binds to the repetitive sequences.